On today's Arabidopsis Research Roundup, I'm delighted to be joined by Professor Robert Sablowski from the John Innes Centre. I'm going to be talking about a new paper just published in Developmental Cell, which has the title of Control of Oriented Tissue Growth Through Repression of Organ Boundary Genes Promotes Stem Morphogenesis. So thank you very much, Robert, for agreeing to do this. And it would be great if you could give us an overview of the paper and the highlights within it.
It's my pleasure. So the background for this paper is that we've been working for a number of years on the shoot meristem, and the listeners will know that the meristem is all plant organs after germination, and therefore is a central focus for plant development. And if anybody reads the reviews on the shoot meristem over a number of years, they will mention the organization of the meristem with stem cells in the center, cells in the peripheral region and organogenesis, and they'll mention the rib zone as the region that produces stem tissues, and then it stops there. The rib zone has been very poorly studied for several reasons. One of them is that it is more difficult to image. It is too deep for good confocal imaging, for example, which has been the basis for a lot of advances in meristem biology. And it is also lacking good anatomical landmarks, even to define precisely what exactly is the rib zone, where it begins and ends. Nevertheless, it is a very important region because we know a lot about flower development, leaf development, and it's all subtended by the stem, which defines the architecture of the plant. It is very important for breeders, for harvest index, for lodging, and all kinds of important agricultural issues. So the starting point for this was that we wished we could understand better what the rib meristem does and how that is regulated, and how eventually that becomes stem with particular morphological features. So we began by focusing on genes that are known to control stem growth, and currently we focus on DELA genes and on Repression, which has multiple other names. It's a homeodomain encoding gene, also known as Pennywise and Repression. We try then to link the activity of those genes to the cellular behavior that actually builds the organs. And to understand how that link occurs, we have to understand how the genes control rates and orientations of cell growth and division and tissue growth. So the first task was to find ways to measure the directions and rates of tissue growth in the rib meristem. And for that we took two approaches. One of them is based on the fact that we have been using a lot a method for confocal imaging of plant tissues in which you covalently link propylium iodides to cell walls. And it happens that the signal in those images is proportional to the amount of carbohydrate in the material. So it follows that thin, weak walls in the images correspond to more recently produced walls, which didn't have time to build up, and those serve as a record of the recent cell divisions without any need to image the tissues over time. So taking advantage of that, we wrote scripts that will analyze the confocal images and derive or detect the most recently deposited walls and then find their orientation. So based on the analysis of the orientation of the most recently produced cell walls, we can infer the directions of recent tissue growth. And we applied that to the rib meristem, and we found something interesting, that it appeared that the regulatory gene Repression had a very significant effect on the direction of cell division and tissue growth in the rib meristem. So in the wild type, there's a very clearly defined central region with mostly longitudinal divisions that feed into the rib zone, and then they're surrounded by cells that divide more often radially that increase the width of the stem. And in the Repression mutant, that was less clearly organized. The distinction between a central zone and a peripheral zone were not as obvious. Then we, of course, needed independent validation of that, and we used another approach to measure the directions and rates of tissue growth, based on genetically marking the cell. So it's a classic approach, sector analysis. In this case, we use the Krylov system to mark cells by GFP expression. And then we analyze in 3 dimensions the main axis and orientation of the sectors in the rib meristem, and found again that in the Repression mutant, the orientations were altered, and again there was a less clear difference between the central part and the peripheral part of the rib meristem. So at this stage, then we concluded that rib meristem function was controlled by Repression in a way that involved the control of orientation of cell division and cell growth. Okay. Importantly, the sector analysis also showed that there was no difference in the rate of cell divisions. That was the same, which was surprising because naively we started with the expectation that whatever controlled the rate of stem growth would control the rate of production of new cells in the rib meristem. That didn't seem to be the case.
Okay.
So that was the result of the cellular analysis. We then, of course, want to know what could be the genetic and molecular basis for these changes. And for that, we used another typical approach. We did ChIP-seq and transcriptomics analysis. And from all that, we ended up with a number of interesting links to other pathways controlling shoot development, such as centroperipheral patterning and adaxial-baxial patterning and vascular differentiation. But one that really stood out was that a very large number of organ boundary genes were directly controlled by Repression. And that rang bells because it had been shown already that Repression interacts with genes that function in organ boundaries, but at a very high level. These are top-level genes that also interact with Repression directly. But now we had evidence that downstream genes more specialized on boundaries were also regulated. And one of those that was very highly regulated and strongly bound by Repression is called LSH4. Okay. That functions downstream of the classic CUC genes, and it turned out to be expressed in the right region. It was around the rib meristem. When we looked at the mutant, the expression of LSH4 invaded the rib meristem, which was suggestive. And finally, we did a key experiment, which was we mutated LSH4 away in a Repression mutant, and that corrected not only the defects in the orientation of cell divisions in the rib zone, but also restored to a large degree stem growth. So we concluded that one of the main causes of defects in stem formation in the Repression mutant was that boundary genes invade the rib zone and alter the orientation of cell divisions, the central peripheral patterning, and we now have to investigate how that unfolds into a change in stem growth. Potentially, it is to do with patterning, which sets up vascular patterning, and we know that vascular differentiation is important for the timing and the degree of stem growth later. So that's one future direction.
Okay, I see. So, well, thank you very much for that comprehensive review of the paper. I've got a question about maybe the start of the experiment, when you first were thinking about how to analyze the rib zone. Because, of course, you have to image through quite a significant number of cell layers from the shoot meristem. So was that, and then, of course, you have to rely on the propidium iodide getting into those cell layers in a consistent manner. So was that a difficult set of experiments to do initially to set up the experimental system?
The experimental system was set up for another purpose, because we have been working on flower development for a number of years, and we established in the lab the method developed by other people to use propidium iodide in cleared tissues to analyze floral buds. Okay. It turns out that using cleared material, the material becomes virtually invisible. It's very transparent. The loss of signal with depth is very, very small. Okay. So we get a very good signal-to-noise ratio quite deep in the tissues. And that's why we decided to apply that to the rib zone, because it goes around some of the initial difficulties in the rib meristem. But there are hidden details. Even though the signal-to-noise ratio is very good, there is a loss with depth, and there is a bias to cell wall orientation in terms of how much signal you get. So the scripts we wrote have to correct for that.
Okay.
So if you dig deeper, you'll find that there are hidden problems that had to be solved.
Yes, no doubt there were. As you said, you did a chip seek, and you talked about the role of LSH1. But did you identify other targets that you're going to work on in the future? You think of the
Yes. Okay. The trouble with chip-seq experiments is that typically they give you too much.
Right. Yeah. Of course.
Yeah. And it's a question of how to prioritize. And in the case of Repression, there are very obvious functional categories that call for attention. And in the case of the Repression work, I mentioned vascular differentiation. That's one of them.
Okay.
Repression directly interacts with virtually everything that has been described to control early vascular development. Mm-hmm. So for example, WOX4 and CLE42 and PIXI, all the receptor ligand systems to control the size of the supply of cells for vascular differentiation, and then later differentiation genes, they are all controlled or at least bound by Repression. So that's an obvious avenue to explore.
I see.
Then we want to understand how boundary genes control tissue growth, and most of what we know comes from work on the surface of the meristem, and that it has been proposed that there is control of cell wall function, cell cycle control, and also auxin responses. Mm-hmm. We have links to all of those through Repression, and we want to explore those links in terms of understanding how tissue growth is controlled, directions and rates.
I see.
So that's another important direction.
Yeah, absolutely. And just one final point. We haven't mentioned the phenotype. So the phenotype of the mutant is that it has a shorter stem. Do you think there's a similar mechanism occurring in other organisms aside from Arabidopsis, so in monocots or in closely related dicots?
Well, the function of the central regulators, such as DELA proteins and also Repression, is known to be conserved.
Okay.
Whether the downstream effectors are conserved, we don't know yet. Okay. But although I cannot say much about conservation beyond the close relatives of Arabidopsis at the moment, related to downstream effects, we know now that the connection to organ boundary genes is important for other genes. We know, for example, that DELA proteins target the same genes, or at least a subset of the same genes. So we expect that will be a hub for control of organ growth, including stem growth, and we will be interested in investigating that beyond the close relatives of Arabidopsis and going to monocots as well, as you mentioned.
Excellent. Well, as ever, the more we find out, the more we need to know. So, So, thank you very much for joining us today, Robert.
It's my pleasure, as I said.
Thanks very much. Cheers.
Cheers.